Hello, Dr. Emmanuel Ugbo. To audience, please wait a minute.
Please, I am coming. Just give me a little time.
Okay, no problem.
Dr. Ugbo, I
Can you
already
see
said you can continue your presentation.
It's okay. Can you hear me?
Okay, perfect.
We hear you.
Good morning, Professor Efinde,
Okay.
Dr. Adiana, and Dr. Primesia, and the other distinguished members there present. Thank you for honoring me to be the guest lecturer in Airlangga postdoctoral program. I will be making a presentation on a comparative mechanism between gram-negative and positive bacteria on antimicrobial resistance. Jamaica, this thing is freezing.
You need to do this now. This works. Just one time.
Why is this thing?
Maybe you should ask them to shoot for you. The assistant lab. Has memories.
Can I escape and start afresh?
Dr. Ugbo, can you see our slide? I'm
Yes.
already share screen, so you can just.
I can see a slide, please. Can you control the slide for me?
Yes, of course.
Okay, okay, that'll be better. So, like I said, I'll be making a presentation on antimicrobial comparative mechanism of gram-negative and gram-positive organism. First of all, in the introduction, the struggle of mankind against infectious disease caused by gram-negative and gram-positive bacteria is well known. The discovery of antimicrobial agents Okay. So the discovery of antimicrobial agents led to optimism that infection caused by controlling infection can be controlled and prevented. It appears that the emergence of antimicrobial resistance is inevitable to almost every new antibiotics or drug, and is recognized as major problem in the treatment of microbial infection in both hospitals and communities. Antimicrobial resistance remains one of the major global health crises and the most serious threat facing human today. Next. Okay. Difference between gram-positive and gram-negative bacteria. Based on their cell wall, gram-positive bacteria has a single layer and they also have a smooth cell wall, whereas gram-negative bacteria has a double layer and has a wax cell wall. Cell thickness. The cell wall thickness of gram-positive bacteria is between 20 to 80 nanometer, and that of gram-negative is between 8 to 10 nanometer. Based on their peptidoglycan layer, it is a thick layer and also can be multi-layered. Whereas in that of gram-negative, it is a thin layer and often a single layered. There is the presence of teichoic acid in gram-positive bacteria, while there is absence of teichoic acid in gram-negative bacteria. Under the gram-positive bacteria, there is absence of outer membrane, but there is presence of outer membrane in the gram-negative bacteria. Next slide. Based on their porine content, there is no presence of porine in a gram-positive bacteria, while under gram-negative bacteria, porine occurs to be present in the outer membrane. Based on their mesosome, it is more predominant or in gram-positive bacteria, but less prominent in gram-negative bacteria. Gram-positive bacteria appears to be cocci and spore-forming rod based on their morphology, whereas that of gram-negative is not spore-forming rod. There is presence of two rings in the basal body of gram-positive bacteria, while there is 4 rings in the basal body of gram-negative bacteria. Based on their lipid content, there is a very low content of lipids in gram-positive bacteria, while the percentage of lipid content in a gram-negative bacteria appears to be between 20 to 30%. And there is absence of lipopolysaccharide in gram-positive bacteria, while there is presence of lipopolysaccharide in gram-negative bacteria. Next slide. Gram-positive bacteria appears to contain exotoxin, while gram-negative bacteria contain both endotoxin and exotoxin. They produce both endotoxin and exotoxin. And based on their resistance to antimicrobial, gram-positive bacteria are more susceptible to a known antibiotics, whereas that of gram-negative bacteria are more resistant to already known antibiotics. An example of gram-positive bacteria is Staphylococcus and Streptococcus, whereas example of gram-negative bacteria are Escherichia coli and Salmonella, etc. And based on their gram staining, gram-positive bacteria retains the primary staining, whereas gram-negative bacteria retains that of the secondary staining. Next slide. Basic anatomy of gram-negative and gram-positive bacterial cell wall. The gram-positive bacteria consists of a cytoplasmic membrane surrounded by a tough and rigid mesh called cell wall. Gram-negative bacteria consist of thin cell wall that is surrounded by second lipid membrane called outer membrane. The space between the outer membrane and cytoplasmic membrane is referred as a periplasma, and the outer membrane is an additional protective layer in a gram-negative bacteria and prevents many substances from entering into the bacterium. Next slide. However, this membrane contains channel called porine, which allows the entry of various molecules, such as drugs. The cell wall is a tough layer that gives a bacteria a characteristic shape and prevents it from osmotic and mechanical stress. The cytoplasmic membrane prevents ions from flowing into and out of the cells and maintains the cytoplasmic and bacterial content in a defined space. Next slide. As you can see the basic structure of gram-positive cell wall and that of gram-negative cell wall. You can see that the gram-positive cell wall appears to have a thick cell wall when compared to that of a gram-negative cell wall. Next slide. Antimicrobial resistance in gram-negative and gram-positive. Antimicrobial resistance in gram-negative and gram-positive bacteria can be of two types. One, intrinsic form and acquired form. Next slide. Under the intrinsic or natural, it's always expressed in the species. That is to tell you that it is being natural. It's not acquired by the organism. It is the innate ability of the bacterium to resist a class of antibiotics. And the most common bacterial mechanism involved in intrinsic resistance are reduced permeability of the outer membrane, that is the lipopolysaccharide, and the natural activity of effluence pump. Next slide. Then, based on acquired resistance, whereby a natural susceptible microorganism acquire a way of not being affected by a drug. That is to say that the organism that naturally susceptible to a particular antibiotic acquires a mechanism whereby they can now form resistance to that particular drug. That is why it is acquired, not natural. Bacteria acquired any genetic material based on transformation, transposition, or conjugation. All are termed horizontal gene transfer. Plus, the bacteria may express mutation to its own chromosomal DNA. And you can see a structure showing transformation, conjugation, and transduction by the different bacteria. Next slide. Antimicrobial resistance. Here you can see what I tried to explain in just the slide that I just finished. You will see that antibiotic resistance is basically classified natural and acquired, and under natural, mostly lack of metabolic processes and targeting sites. That's what they can actually cause the resistance. But under acquired, that's where you talk about biochemical mechanism and genetic method. Under genetic method, that's where you talk about mutation. Mutation can actually cause resistance. When an organism mutates, it can actually bring up a gene that can actually cause resistance. And the next one is acquisition of extra chromosomal DNA called plasmid, can equally cause resistance in a particular bacteria. And based on the biochemical mechanism, one is production of enzyme that prevents drugs accumulation, and modification of target site. That is, the drug target site can actually be modified by a particular organism, or use of alternative pathway, and also quorum sensing. Next slide. Here are the mechanism of action of antibiotics. You can see the cell wall synthesis. For example, the beta-lactam antibiotics and the vancomycin antibiotics, and the nucleic acid synthesis, where you now talk about the DNA gyrase, the quinolone, and the RNA polymerase, that is the rifapin, and the protein synthesis, where you talk about the microlith and so on, and the one that attack the protein synthesis of 30S. You talk about aminoglycoside and tetracycline. These are the possible drugs and their target sites where they can actually target, the site they can target in the bacteria cell. Next slide. Mechanism of antimicrobial resistance. Bacteria develop antimicrobial resistance by several mechanisms. One is limit uptake of drugs, that is natural. And secondly, modification of drug target site. Thirdly, inactivation of the drug and active effluence pump of the drug. Like if you look at the structure of the bacteria that was displayed, you can actually see the effluence pump and the antibiotic degrading enzyme and so on. Next slide. Mechanism of antimicrobial resistance continues. Because of difference in structure, there is a variation in types of mechanism used by gram-negative bacteria versus that of gram-positive bacteria. Gram-negative bacteria make use of all the four mechanisms of resistance, whereas gram-positive bacteria less commonly use limiting the uptake of drug. It does not have the lipopolysaccharide outer membrane and does not have the capacity to contain types of drug effluence mechanism that can be applied. Next slide. Here is the mechanism of antimicrobial resistance. Here you can see the impermeability, the modified cell wall protein, whereby the organism have actually modified the cell wall in the sense that when a drug target comes, it cannot actually target it and it will not penetrate into the cell wall because the cell wall of the bacteria has been modified. And next one is acquisition of plasmid. You can see the presence of plasmid that can contain one gene or the other that can actually harbor resistance. And the second one is the modification of drug target site. You can see the drug target site for the second column of this bacteria has been modified in the sense the drug cannot actually cook into the target site because of the slight modification of the target site. Thereby, the organism, the drug will not be able to inhibit the activities of this organism. Next one is the effluence pump. Increasing activity effluence of the drug. You can see when the drug gets into the bacterial cell, it now uses effluence pump to push the drug out of the cell, thereby making the drug inactive to inhibit the growth of the bacteria. The last is the inactivation. Inactivation means the addition of a phosphor group on the antibiotics, which will reduce the ability to bind to the bacterial ribosome. Next slide. Yes, we are going to take it one by one to explain the 4 different mechanisms utilized by both gram-negative and gram-positive bacteria. 1, limit uptake of a drug. This is a natural differences in the ability. There is a natural differences in the ability of bacteria to limit the uptake of antibiotic agents. The structure and the function of LPS layer in gram-negative bacteria provides a barrier to a certain type of molecules. Next slide. Certain bacteria modifies their cell membrane porine channel, thereby preventing the antimicrobial from entering into the cell wall. That is genetical. This one can actually appear in form of genetical. It's not natural. There are 2 main ways in which porine change can limit uptake, drug uptake. 1, a decrease in the number of porine presence. And the second one is mutation that changes the selectivity of the porine channel. Next slide. This strategy has been observed in a gram-negative bacteria such as Pseudomonas, Enterobacter, Klebsiella species against drugs such as imipenem, aminoglycoside, and quinolone. Next slide. Gram-positive bacteria such as Staph aureus recently has developed resistance to vancomycin. Of the 2 mechanisms that Staph aureus uses against vancomycin, 1, Staph aureus produces a thickened cell wall, which makes it difficult for the drug to enter into the cell and provides an intermediate resistance to vancomycin. These strains are designated as Visa strain. That is, they are non-genetical. Next slide. Second mechanism, modification of drug target site. There are multiple components in bacterial cell that may be targeted or that may be the targets of antimicrobial agent. There are just as many targets that may be modified by bacteria to enable resistance to this antibiotic or drugs. Next slide. One mechanism of resistance to beta-lactam drugs used almost extensively by gram-positive bacteria is via alteration in the structure and a number of penicillin binding proteins. Penicillin binding proteins are transpeptidases involved in the construction of peptidoglycan in the cell wall of a bacteria. Next slide. A change in number, that is increase in peptidoglycan, increase in penicillin binding proteins that have a decrease in drug binding ability, or decrease in penicillin binding protein with a normal drug binding of PBS impacts the amount of drug that can bind to the target. That is to tell you that when there is an increase, when the bacteria produces increased number of penicillin binding protein, the number of drug that we bind will not be enough to bind to all the site of the PBS, thereby causing resistance to that particular antibiotic. Next slide. A change in structure. That is when the PBS has changed its structure to PBP2A. For example, in Staph aureus, by acquisition of a MEK gene, may decrease the ability of the drug to bind or totally inhibit drug binding. That is to say that when there is a modification in the structure of the PBS, thereby producing PBP2A, which is commonly found in Staph aureus, which causes the acquisition of MEK gene in Staph aureus. Next slide. The glycopeptides, for example, vancomycin, also work by inhibiting cell wall synthesis, and lipopeptide, example, daptomycin, works by depolarizing the cell membrane. Resistance to vancomycin has become a major issue in enterococci. For example, vancomycin resistant enterococci, or in Staph aureus, methicillin resistant Staph aureus. Next slide. Resistance is mediated through acquisition of van gene, which results in the change in the structure of peptidoglycan precursor that causes a decrease in the binding ability of vancomycin. And under the daptomycin requires the presence of calcium for binding. Mutation in the gene, example, MPRF, changes the charge of the cell membrane surface to positive, inhibiting the binding of the calcium and other daptomycins. Next slide. Resistance to drug that targets the ribosomal subunit may occur via ribosomal mutation. For example, in aminoglycoside, ribosomal subunit methylation most commonly involve ERM gene or ribosomal protection, for example, in tetracycline antibiotics. And this mechanism interferes the ability of the drug to bind to the ribosome. Next slide. For drugs that target nucleic acid synthesis, for example, fluoroquinolones, resistance is via modification in the DNA gyrase. For example, in gram-positive bacteria, that is the production of gyra A or topoisomerase 4 in gram-positive bacteria, which gives to GRLA. The mutation causes change. These mutations causes changes in the structure of gyrase and topoisomerism, which decreases or eliminates the ability of the drug to bind to these components. Next slide. Inactivation of drugs. There are two main ways in which bacteria inactivate a drug. One, actual degradation of the drug or transformation of a chemical group to the transfer of chemical group to the drug. Next slide. Actual degradation of drug. The beta-lactamase are a very large group of drug hydrolyzing enzyme. Another drug that can be inactivated by hydrolysis is the tetracycline via the TETX gene. Next slide. Transfer of chemical to the drugs. Drug inactivation by transfer of a chemical group to the drug most commonly uses a transfer of acetyl group. There are a large number of transferases that have been identified. Acetylization is the most diversely used mechanism. That is to say that the acetylization is the most diversely used mechanism and is known to be used against the aminoglycosides. Next slide. Active effluence of a drug. Bacteria possesses chromosomal encoding gene for effluence pumps. Some are expressed constitutively and others are induced or overexpressed. High level of resistance is usually via a mutation that modifies the transport channel. Under certain environmental stimuli and when a suitable substrate is present. The effluence pump functions primarily to rid the bacterial cell of toxic substances, and many of these pumps will transport a large varieties of compounds. Example, multi-drug and effluence pumps. Next slide. Most bacteria possesses many different types of effluence pump. There are 5 main family of effluence pump in bacteria, classified based on their structure and energy source. 1, the ATP binding cassette family, the multi-drug and toxic compound extrusion family, and the small multi-drug resistance family, the major facilitator super family, and the resistant nodulation cell division. Next slide. Effluence pump found in gram-positive bacteria may confer intrinsic resistance because of being encoded on the chromosome. That is natural. There are also gram-positive effluence pump known to be carried on a plasmid, that is acquired one. These pumps include members of the MEX and the MFS family. Whereas effluence pump found in gram-positive bacteria are widely distributed and may come from all 5 of the family, with the most clinically significant pump belonging to RND family. Next slide. Yes. Here is the comparative or comparing of the resistant mechanism of gram-negative and gram-positive bacteria to antimicrobial agent. You can see antibiotics classes, resistant type, and resistant mechanism, and the common example of bacteria that can actually possess such type of mechanism. For example, under aminoglycoside, the resistant type that you can see is decreased uptake enzymatic modification, and based on their mechanism of resistance, it is occur by changes in the outer membrane. An example of organism that can make use of that mechanism is Pseudomonas aeruginosa. Then under beta-lactam, altered penicillin binding protein and enzymatic degradation. This is whereby the penicillin binding protein is being changed to PBP2A, and it is common in MEC-A in S. aureus. The next one is glycopeptide, altered target. Here the D-anilate alanine is changed to D-aniline D-lactate. And this is common in vancomycin resistant in Enterococcus family. Next one is under microliths. The resistant type is targeted altered target site. And under this altered target site, the resistant mechanism is ventilation of a ribosomal active site with reduced binding. And example is found in ERM encoding the ventilation in the Staph aureus, and also in Pseudomonas, in Streptococcus pneumonia and Streptococcus pyogenes. And another one is they can equally use the effluous drug pump. And next one is under the quinolone group, they use altered target site, and the mechanism is based on mutation that leads to reduced binding to active sites. And they can equally use effluent pump, where they use the membrane transporters to transport out the drug from the cell of the bacteria. And this is common in mutation in gyrase A in enteric gram-negative bacteria and gram-positive bacteria such as Staph aureus. Another one is the tetracycline antibiotics. They make use of effluent pump and also altered target site. New membrane transportation or transporter is the mechanism of resistance that they possesses, and production of protein that binds to the ribosome and alter the conformation of the active site. And it is common in TET gene encoding effluent protein in gram-positive and gram-negative bacteria. Next one is the chlorophenicol. Chlorophenicol make use of resistant type called antibiotic inactivation and effluent drug pump, and also alter the target site. Here, the resistant mechanism is by chlorophenicol acetyl transferase, is an enzyme, and it is common in Streptococcus pneumonia. Next slide. Here is the summary of antibiotic resistant mechanism in important gram-positive bacteria. It's just almost the same thing with what I explained in this slide that I just finished. Here is just trying to show you the different types of antibiotics and the possible enzyme that can actually cause the resistance. And here I was able to get information on Staph aureus and Enterococcus. As you go down, you can see it's just almost the same thing with what I explained in the other side. Next slide. The same thing applicable here. Summary of antibiotics resistant mechanism in gram-negative bacteria. Like here, we have penicillin. Under penicillin, Pseudomonas aeruginosa produces AMP-C enzyme and also extended spectrum beta lactamase and other beta lactamases, which they can use to inhibit the activity of these antibiotics. The same thing is applicable in E. coli. As you go down, you can equally see, like under this fluoroquinolone, the Pseudomonas aeruginosa uses mutation in the topoisomerase 4 and DNA gyrase gene, and they equally make use of effluence pump, and those are the things they can use to inhibit the activity of the drug. And under the E. coli, they use mutation in the DNA gyrase gene. The same thing with other antibiotics that are being applied. Take for instance, aminoglycoside. Under aminoglycoside, Pseudomonas aeruginosa makes use of aminoglycoside degrading enzyme and also effluence drug pump, whereas E. coli makes use of amino acid degrading enzyme just to use to degrade the drug, to make the drug inactive. Next slide. In conclusion, gram-negative bacteria are more resistant to antimicrobial agents because of the ability to make use of the four known mechanisms of resistance, whereas gram-positive bacteria are less resistant to antimicrobial agents because they do not utilize all the four mechanisms of resistance. Next slide. Thank you for being part of this lecture. And I don't know how to start. It seems that I apologize every day. I don't know. It seems my system is having a little issue. I don't know. Whenever I try to share my slide, it keeps on hanging. So apology for that, please. So I want to use this opportunity to thank my hosts, Professor Efindi, and the faculty international representative, Dr. Adriana, and the chief host, the person that is anchoring this particular guest lecture, Dr. Prima, and other distinguished members that we are present for this lecture. It is my privilege to be part of you people, and I am very glad to have been affected by you people positively. And I equally hope that one day after the pandemic, we'll be able to meet in person by special grace of God. Thank you for
I'm waiting for you, Dr. Ugbo. Please come to my department.
Yes, I equally want to thank Department of Public Veterinary Health.
Public Health.
And Department of Public Health, Faculty of Veterinary Medicine, for finding me worthy to be one of their postdoc students. I am humbled to be part of you. Thank you so much. And
you are welcome, Dr. Ugbo. You are welcome.
Thank you, sir.
You are welcome.
Okay, you're welcome, Dr. Ugbo. That's the lecture from Dr. Emmanuel Nobuki Ugbo. So, ladies and gentlemen, we are now going to take question and answer session. For the audience, you can ask directly to Dr. Emmanuel, but please mention your name, please. Yes. Maybe in the chat box, Dr. Amir.
Oh, Amir Session.
Amir. Yeah. There are questions for Dr. Amir. My question is that sometimes we have good result in the lab, but when we use trial in the animal, it doesn't give us a good result. Why? Dr. Emmanuel, maybe you can answer directly for this question.
Okay. Okay. You said sometimes we have a good result in the lab, but when we try it on animal, it does not give a good result. Well, my own little knowledge based on that is that sometimes maybe take for instance, you are trying to kind of check if an organism is resistant or can cause infection in that can, or maybe you are able to get a particular organism that you have actually certified in the laboratory that it is capable of causing infection. And when you try it on animal, it may not be able to cause the said infection you expected it to cause. The result could be because of the immune system of that particular animal, because sometimes our immune system also contributes to the level of susceptibility we can be to a particular infection. Assuming that two people are living in a particular place and the mode of feeding might not be the same thing. Your like and my dislike might not be the same thing. I may feed on things that give me more immunity, and you can feed on things that just give you more sweet on your mouth. So at the end, you see, the one that feed on something that protects him more tends to have a higher immune system than the ones that feed on things that just gets him satisfied. So sometimes the result could be as a result of the hard immunity of that particular animal. I don't know whether I'm able to make an input.
Yeah, thank you. Yeah, I have understand, because we do the lab work when we do the mastitis cases, mostly in streptococcus. We found very difficult when we got the result of oxy or gentamicin or streptomycin. But in practical, when we use in animals, we do not get the same results which we are getting in the lab.
Okay, still the same thing Dr. Prima is saying, that sometimes you get a good result in the lab, but when you try it on the animal, it doesn't give a good result. I don't know. I don't know. Like, all I can say is still what I said before. That's the little knowledge I know about it. And at times, it could be the mode of inoculation. It depends on how it is being inoculated. Because, you know, sometimes when we inoculate, either maybe when you inoculate intravenous, you inoculate something intravenous, it tends to go faster than when you inoculate intradermal or intrathecal. I don't know whether you are getting what I'm saying. So because the intravenous gets straight to the blood, but the other ones, it tends to take time before it will start to assimilate into the blood. So it could be the mode of inoculation, because I can promise you that if you inoculate intradermal or intramuscular, before the substance will get to the blood, it might have dissociated to a certain level. Do you understand? Compared to when you inoculated intravenous, it will go faster under intravenous than when you inoculate intramuscular. Thank you.
Thank you very much.
You're welcome.
Okay. And then the next question from Dava about amoxicillin. Maybe you could answer this question about amoxicillin. Amoxicillin is an example for antibiotic that is very easy to get with a doctor prescription, right? And the risk of this amoxicillin resistance could be greater, consider the massive use and with the doctor prescription. However, this drug is still option for the treatment. So the question is, there no amoxicillin resistant in bacteria? If not, how can it happen?
Well, there is no antibiotics that does not have an organism that resists it. There is none. If there is, maybe there are the newly produced antibiotics that are not being widely used. But the major thing is that there are some drugs that intends to be more effective. Even at a hard period, they are still assumed to be the best option in treatment of infection. Like amoxicillin, there are a lot of organisms that resist amoxicillin, and there are a lot of infections that are being treated with amoxicillin. But you know, there is another thing about drug resistance. Like those drugs that intend to be more available in the counter, that you can get in tablet form or in a syrup form, intends to pose the microorganism intends to have more resistance to those drugs than those ones that appears in injection, because those ones that appears in injection are not much abused. Do you understand? They are not much abused because it's just a regulated number of people that uses it to treat an infection. But those ones that appears in tablet or in capsule is easily abused. So to me, there is always a resistance in every antibiotics that has been widely used. For example, like that of amoxicillin. Thank you. I don't know whether I'm able to make an input in your question.
Okay, Dafase, thank you, sir. And Mr. Amir, there is another question for Mr. Amir. What is cause AMR in your country? The cause of AMR in your country?
Yes, in my country, the cause of antimicrobial resistance mostly in the meat and drug, and meat and chicken in my country. You know, my country, Nigeria, is a multi-ethnic country. Do you understand? So, and I can tell you that it's the biggest country in Africa. We have a lot of population, and you know, as a result of a shortage of food in the whole world, so people intend to get result quicker, to brood animals faster, and as a result of that, trying to brood animal like chicken and cow and so many other things, they intend to use most of these drugs that are used in form of growth promoter. They use it in promoting the growth of the animal, whereby animal that are supposed to take like 5 months to be a standard animal, to be to the level that it can be consumable, can equally use like within 2 months to 3 months to be enough for consumable. You know, by so doing that, you have overused the antibiotics in form of amplifying the growth of the animal. And by so doing, a time will come, this particular antibiotics you are using to amplify to use as a growth promoter can no longer function very well as in terms of treatment value. Do you understand? So all those abuse can actually lead to antibiotic resistance. And also, people intend to treat their animals like chicken and cow without the knowledge of vet doctors. Because in a normal setting, you don't have any right to administer any drug to animal without a consultation of a vet doctor, but people abuse it here. So that's one of the major cause of antimicrobial resistance in my country. Thank you so much.
Okay, this is the answer from Dr. Amir. The next question for Dr. Saifur about the drug of choice in mastitis. What the drug of choice of mastitis? And the second question, if we use the milk that animal after injecting antibiotic, it's safe or not?
Please, can you come again?
Oh, I'm sorry.
Yes.
The first question about drug of choice in mastitis. What the drug of choice in mastitis?
Mastitis, you mean?
Mastitis.
Yes, I
Yes.
can hear you. Yes. Well,
Thank you, Prof.
इकट्ठे लड़ो आप यार ना
It is very country to country. In my country, in Pakistan, due to streptococcus and staphylococcus, mostly we use norfloxacin, enrofloxacin, streptomycin, gentamicin, like that, and ciprofloxacin as well.
No.
Yes.
Yes, also vary from country to country, but overall, according to OI rule, only one or two drug of a choice for mastitis or any other disease like that.
Okay.
In mastitis, we use many antibiotic like gentamicin, streptomycin, sometimes sulfadamidine also used in mastitis and other drugs used. But what is a drug of a choice in mastitis we can use in everywhere all over the world?
Thank you, Doc. You know, in gentamicin, you know, if you have worked with gentamicin for a very long period of time, you will see that gentamicin appears to give more good results in terms of treatment value. Or even when you use it to test for resistance, it tends to the organism tends to be susceptible to gentamicin. The reason behind this is because gentamicin appears to be more in injection. Do you understand? It appears to be injection. So it is not in a tablet or in a capsule form. So it has not been widely abused. People don't intend to lay their hand easily on it because you must apply it through injection. Thank you.
Second question, according to a route of injection or route of entry of a drug, in mastitis, we use intramammary or intramuscular, or which route important for injecting the antibiotic, so we get result of treatment as soon as possible.
Okay, you know, when you are to inject antibiotics, every mode of injection has its own side effects. I don't know whether you know that. Take for instance, intramuscular, it has its own effect. It can cause tissuing or any other thing. Then that of intravenous. Intravenous, if you use intravenous, you can only use intravenous in a serious case. Take for instance, if the case the animal is having, you've looked at the animal and you know that if you are to start treatment with intramuscular, you cannot achieve the valued result you want to get. That will be the time you can now use intravenous.
Yeah. No. In case of mastitis, I have a question. In case of mastitis.
Okay. What is the question?
Yeah, question. We best route we use. We inject the antibiotic or any other sportive drug intramammary or through intramuscular or through intravenous. Which is the best route? so we get result as soon as possible.
Well, if you want to get results as soon as possible,
yes,
the one you will use is intravenous. If you use intravenous, you will get results as soon as possible because blood circulates every part of your body. Do you understand? But
yes.
because this mastitis, the infection we are talking about, are being circulated by the blood. Do you understand? The organism, these organisms are being carried by the blood. So when you inject intravenous, it picks it very fast than when you inject intramuscular or intrathecal or any other means. Thank you.
Okay, I want to little help you in this question. When we are using the drug for mastitis, we keep it in our mind that how much residues come in the milk for mastitis. So first of all, for example, when we are using the gentamicin, as doctor asked, we use mostly gentamicin intramuscular so that it releases slowly because always streptococcus and staphylococcus needs time. And also on the other side, we also use this gentamicin as a intramammary tube as well. So we use in mastitis cases both routes, intramuscular as well as intramammary. So I want to add this. Thank you very much.
About what your opinion, dear doctor, about the milk of mastitis animals like cattle, like bovine, like buff, or like any other animal. Can we use the milk of that animal? Is it okay for the health or not? Can you understand?
Okay, I understand. You are trying to know if animal that is suffering from mastitis, if you can use the milk. Is it not what you're asking me?
Yeah, yeah.
If the milk can be used. Well, you know, we are talking about public health here. Do you understand? So it is better if the animal is treated before you can take the milk, because the major agent that is causing the mastitis can actually be found in the milk. Because if you have gone through some research has published that you can actually find a traces of resistant microorganism in milk gotten from cow or any other milk producing animal. So for my own suggestion is it is better it is treated first before you now plow or milk the animal. Thank you.
So according to your opinion, we cannot use the milk of mastitis animal. Yes. If we boil the milk, at 95 degree, then it is possible the bacteria like staphylococcus or streptococcus, they kill like that.
Yes.
Heat treatment, through heat treatment.
Yes. You can use it like I want to make my point clear here. So if it is raw, it is not good to be used. If you are using it raw, it is not good to be used. But if you know that you milk it and it will pass through treatment, and the treatment is certified that it can kill the presence of the organism that is causing the mastitis, that is good enough.
No, through pasteurization, we can use the milk of mastitis animal, yes?
Yes. You can use it. Yes, you can use it.
Thank you. So through pasteurization, if we use the antibiotic in the mastitis animal or any other animal, we use the antibiotic, so we can use also the milk of that animal.
Yes. Once the animal has started undergoing treatment and you are certified that you can achieve 100% pasteurization, that if you pasteurize the milk, you will do proper pasteurization in the sense that there will be no presence of a microorganism. You can use it.
So
it is usable.
Yes. According to milk and other, I have another question in my mind. Mostly in the villages, everywhere in the world, people use milk without boiling. They cannot boil the milk. According to your opinion, we can use the unboiled milk. Is it okay? Or if we boil it, its quality and its protein values remain same or change?
You know, when you boil a milk, you know, milk contain a protein content which you can denature when you pass it through heat, unless on a regulated heat like pasteurization. Do you understand?
Yes.
You know, pasteurization is a regulated heat treatment of milk.
Yes,
but if you do local boiling of a milk, just an ordinary heat boiling, there is every tendency that you can break the protein content of that particular milk, whereby at the end, what you are taking is chaff. It's no longer giving you any nutritional value. You have already denatured the nutritional value. So that's why it is not all that advisable to boil your milk. To boil your milk unless you are boiling it under a control condition that has been a certified condition for treatment of milk.
Last.
Okay.
What is the main reason most of the zoonotic diseases come from the continent of Africa? What is the main reason?
Okay. Well, it's not really that zoonotic infection is mostly.
Yeah, if you see the history, like Ebola, like chickenpox, like Congo, and other mostly diseases come from the Africa. What's the main reason?
Okay.
Either it's the climatic change, or the living status of the people, or the culture. What's the main reason?
The problem is that in Africa, you know, Africa is an underdeveloped country. Africa is underdeveloped country. Do you understand? And sometimes we have in Africa, we have a lot of leadership problem. We have a lot of leadership problem whereby you see people are categorized into different value, high class, middle class, low class, and blah, 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 blah. You know, yes, that kind of thing. So this thing now intends to make those people that are coming from remote areas, most of them intend to live in a forest with their animals. Do you understand? Like now in Africa, we have what we call herdsmen, people that live with animals in forest. Most of them live their life, all their life in the forest with animal. Do you understand? So, you know, when someone is living and sleeping in the forest without shelter, when there is rain, there are so many things. Those are the kind of people that sometimes they can acquire one disease from the forest with their animal. When they come to town, they can actually transmit it. Or when they get to their village, they can transmit it. So the problem of most of the zoonotic infection coming from the Africa is because of the life pattern of Africans with their animals in the bush, unlike in a developed country. Like in a developed country, they do what is called ranching. Do you understand? They do what is called ranching. They do ranching of their animal in the forest, where they will build just a mini standard company, where they will equally be cultivating grasses and employ people. People will be living there and not living in the forest. Do you understand?
Yes, sir. So
they will control and equally vaccinate their animal time to time. But here, we have a problem of all those vaccination or no vaccination because of increase in population, one. And there is no much availability of things for people to take good care of themselves. Unlike in a developing country or developed country, whereby certain values are being given to human life.
Yes.
Thank you.
Yeah, WHO has more focus on African countries, but still the diseases come from the zoonotic diseases. That is the main reason you have explained. Thank you very much.
You're welcome.
Sam, finish or have?
Thank you, Dr. Saifur. I think the question and answer session is enough. Thanks to the audience that had participated actively. And then for recapitulate the message for our lecturer, the first, antimicrobial agent can control and prevent infectious disease that caused by gram-negative and gram-positive bacteria. Antibiotic resistant, gram-negative and positive mechanism by natural and acquired mechanism, bacteria modified cell wall protein, modified drug target, increase active efflux of drug and reduce drug ability to bind the bacteria ribosome. So I say thank you to our speaker today, Dr. Emmanuel, and everyone for attending today's guest lecture. And now I would like to hand over the certificate to Dr. Emmanuel. This is the certificate will be handed over virtually. Thank you, Dr. Emmanuel.
Thank you.
You're welcome. Finally, we get the last session that is closing. On behalf of committee, I would like to thank to the speaker and all the participants for participation today. If there are any imperfection during the implementation of this event, I like to say apologize to everyone. So let's close this event by saying Alhamdulillah. Alhamdulillah. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.
Salam. See you. Thank you very much, Dr. Ugbo.
Okay, thank you, Dr. Ugbo. You can leave the meeting Zoom.
Thanks.
Please come to Indonesia, doctor. Please, we are waiting for you.
It's okay, Prof.
Nice to hear you, doctor. Nice to hear you.
Thank you. Thank you.
Thank you very much.
Thank you. Thank you,
Okay. Dr. Adiana, see you then.
See you, Dr. Ugbo. See you. Thank you, Dr. Wiwi. Thank you, Prof. Wiji. Thank you, Dr. Amir. Thank you, Dr. Saifur.